Oh, thank you very much. Thanks for having me here today. And before I start, I'd like to thank Lori Abity and Eric Hoagland, who are folks within the Human Research Program who know a lot more about Artemis than I do and provided a lot of great information for me to make this presentation possible. So thank you to them. Next slide, please. So the Human Research Program has a mission to enable space exploration beyond low Earth orbit by reducing the risks to human health and performance. Next slide, please. And we do this by focusing on three specific goals. The first is to perform the research necessary to both understand and then reduce those risks. We have about 30 identified risks that are in the books, and I'll show those to you in just a moment. And we work on understanding those. It's not quite basic research like Lisa was talking about. We are an applied research program. But some mechanistic work and just understanding how is this affecting the human. We do a lot of translational work from the animal to the human or from the cell. And then we also enable development of spaceflight medical and human performance standards. These are things like radiation exposure limits, nutritional requirements, EVA requirements, all those types of things. We work with medical operations on those. And then finally, we develop and validate technologies that we can use to both characterize and mitigate those risks. The best example here is the ultrasound machine that's currently aboard the International Space Station. So that machine has had quite a progress through the years. When we first flew the International Space Station, it was the size of an entire rack, literally the size of a washing machine, very heavy, very cumbersome. And you were pretty much locked into that one spot on station. You couldn't move around. The current ultrasound machine is a laptop with a probe attached, and the future ultrasound machines are going to look just like this. They are going to be cell phones, and they're going to have probes attached. So making that transition, miniaturizing the devices, validating them, making sure they work in microgravity is another thing that we do. Next slide, please. It's important to understand the different hazards that are associated with spaceflight and how they interact with different types of spaceflight. And we've identified 5 hazards to the human. The first one is space radiation. This one's relatively obvious. People understand what this means, right? It's the radiation coming from galactic cosmic rays for one, and solar particle events for another. Every time I mention radiation, people automatically assume cancer, and that is one of the risks that we study. But we also study the effects of radiation on the central nervous system, as well as the cardiovascular system. Isolation and confinement is another. You're away from your family. You're in a very small space with, depending on which vehicle you're on, 2 to 4 or more of your closest friends. And when you're in low Earth orbit, you see a sunrise every 90 minutes. So sleep loss, your circadian rhythm is disrupted. Team performance, how do you interact with people, as well as behavioral medical performance. Do you feel depressed? Do you feel anxious? These types of things, they all are affected by the type of environment you're in. Just imagine back when we were all isolated for COVID and you couldn't really leave your house, what that felt like. And, your house is a whole lot bigger than any of the vehicles we plan on taking to the moon or Mars. Distance from Earth is another. So low Earth orbit, this is relatively a small hazard, but think about going to Mars. This is going to be a much greater hazard. And right now, if we have a major medical emergency, we can relatively easily evacuate them from the space station back here to Houston. This will become impossible as we go to the moon and Mars. So we need to figure out how that autonomy plays into all of these risks. Communication delays are an important part of it, and just-in-time training, all these types of things. The fourth one is gravity or lack thereof. This is the main one that I've spent my career studying, and how that affects the human physiology, whether it's the cardiovascular system, bone, muscle. immune system, all these other things are greatly affected by the gravitational field you're in. We've evolved here on Earth in 1G. Now you go up into microgravity where you have this perceived lack of gravity, although the physicists will tell you we still have plenty of gravity there, but you're in free fall, so you have this perception of microgravity. Now, as we go to the lunar surface with 1/6G and the Martian surface at 3/8G, how does that affect the physiology? Is it closer to microgravity? Is it closer to 1G? And what does the shape of that curve look like? We don't know the shape of that curve. And when you talk to different space life scientists and you ask them to draw you the shape of the curve, you get many, many, many different shaped curves. And so understanding this is a key part for going to the moon and Mars. And then finally, hostile closed environments. Um, you're in essentially a tin can up in space, and how does that affect you? You can be exposed to things. There's quite a microbiome of the built environment, the noise, all these other things affect the human as well. Next slide. And so we do our research in a stepwise approach. We do as much as we can here on Earth, and we can simulate lots of different parts of the spaceflight experience with ground analogs like the NBHAB, which is a bed rest analog. We have multiple experiments in Antarctic stations. We have isolation facilities and a space radiation lab. Now in low Earth orbit, we can do even more because then we can introduce the true microgravity component, which is very, very difficult to simulate here on Earth. And we can use that as an analog for going to moon as well as for going to Mars. And then the lunar missions, oops, can we go back for just one sec? And then for the lunar missions, we will heavily rely on the lunar surface to replicate as close as we can to the Martian surface. So we're going from one partial G environment to another, from 1/6 to 3/8. It's going to be a great place for us to do a lot of research. Okay, next slide. I mentioned our risks, and here's a list of them just to give you an idea. So on the far left, that's a listing of the 30 or so risks that we have split up by hazard, and you can see the different types of missions that we've defined here in these columns that are so colorful as we move to the right. So green means the risk is low, yellow means it's a moderate risk, and red means it's a high level risk, something that we need to mitigate. And as we move to the right, we're getting further away from Earth and longer duration. And you can see the two that I've highlighted are for lunar orbital with surface ops, and then Mars, because that's what we're talking about here today. And so this is what the risk profile looks like. You can see we have a lot of yellow and red for lunar and even more red for Martian. So this is where we're spending a lot of our resources. Next slide. And pardon the cat walking by, because the cats own the house, not me. And here's where we do our research, the International Space Station, the MBHAB that I mentioned, a great bed rest facility, NSRL, NASA Space Radiation Laboratory at Brookhaven National Labs. Hera is our short duration isolation analog. We also do parabolic flights. Lisa mentioned parabolic flights. We sponsor those missions as well. We have 2 experiments ongoing in different places in Antarctica, and then we do have some long duration isolation analogs like the NEK as well. Next slide. And we do partner with lots of folks. Here's just a quick list. We work very closely with BPS, as Lisa mentioned. We also work with other NASA programs and groups. We also work with international folks like ESA and DLR and those folks, domestic OGAs as well as industries. We're heavily involved in the commercial spaceflight program. We're working on the CLDs and lots of other things as well. So we're dipping our toes in as many places as humanly possible. Next slide, please. And yes, the pun was intentional. Here's just a little graphic that we all realize, but sometimes you just need to see it. As we get to Moon and Mars, the mission constraints go up, which by necessity makes the research opportunities go down. A lot of what we're trying to do on the moon is bring back rocks and do that kind of work. So there isn't a lot of mass, up mass or down mass for us to do human research. And so this does constrain the types of work that we're going to be able to do. And I really want to talk to Lisa about the LEAF study, really interested in that. And I want to see if there's some way that we can get involved in that as well. Next slide. And so I mentioned that we're involved in Artemis. This is where Lori and Eric do a lot of their work. We didn't do that much with Artemis 1. We took advantage of some things going on, but we do have science on Artemis 2, and we're planning science as we go forward. The further into the Artemis program we go, the more science we plan on doing. We are again having to modify our science because of the constraints of the mission. So instead of bringing back blood samples, we're going to be using dried blood spots and saliva cards and things like that. But we have all of this work that we're working on now, trying to figure out how we do it, who we integrate with, and what is possible to go forward. Next slide. Lots of reasons on why to do Artemis. It's not just because it's new and cool, but the radiation on the planetary surface is going to be different. Isolation and confinement, again, as you can imagine, is going to be different. We're going to have a rover. We're going to have all these other opportunities, and it's just going to be different than being on a relatively large space station. People think it's small, but in the grand scheme of things, it's quite big. Distance from Earth, we're going to have comm delays and things like that, even on the lunar surface. And we need to figure out how to work with that. Again, alter gravity, being on the lunar surface 1.6G instead of microgravity. Is 1.6G enough to act as a countermeasure like it would be coming back to Earth? Or is it going to be a continuation of the changes we see in microgravity? And does the rate change because you have that partial gravity environment? We just don't know. And then finally, hostile closed environments, which may also be some hostile open environments as well. So again, a challenge there. Next slide. Just to give you an idea of some of the types of things we're looking to do on the lunar surface. Now, these are lunar surface only. We also have different projects going on for Gateway and for transit and other things like that. But for lunar surface, we have a sensory motor assessment for pre-EVA. This will include some cardiovascular monitoring, ultrasounding, balance testing, videos, and some surveys. Manual control in-flight training. This will be important for landing on the lunar surface potentially, and so involves hand controller, some simulation software with some data collection. We have what's called standard measures that we've been doing on station and in bed rest for quite some time. We're going to adapt those for Artemis as well. There will be research on lunar descent and ascent, looking at injury assessment. This is something that we've been concerned with here on Earth. And since there won't be a large contingent of physicians available on the lunar surface when the crew lands, we need to make sure we thoroughly understand this. Immune biomarkers, I mentioned the dry saliva booklets as well as the dry blood spots, looking at various biomarkers over the course of the mission, how they're affected by radiation and stress in the environment on the lunar surface as well as transit. And then finally, alterations of multisensory integration. This involves things like dosimetry and different videos where we can see the crew interact and behave. Next slide. Some things for kind of far out in the future. This is more visioning than really telling you what we're going to do, but possible payloads we could work on, and many of these would be in collaboration with space biology, are things like tissue chips, looking at pharmaceutical stability on the lunar surface, plants. Lisa told you a little bit about some plant growth. We're especially interested in plants as crops and nutritional capabilities of those plants. Dosimetry, how is that on the surface compared to being in orbit? And then potentially some type of analytical laboratory where we could do sample analysis right there on the lunar surface and not have to try and send samples back to Earth to be analyzed. Next slide. And finally, just to wrap everything up, so the Human Research Program is dedicated to discovering the methods and technologies to support safe and productive human spaceflight. We intend to use the lunar surface, lunar transit, everything as analogs to get us to Mars. And by conducting human research on the early lunar surface crews, we hope that will help us not just for Mars, but also for later Artemis missions when we intend to stay there for a greater length of time. And that's all I have. Thank you all very much. It was great talking to you today, and I will be sticking around for questions later as well as the panel discussion. So thank you all very much.